Recently, I attended a trip through PDS to Princeton University's Neuroscience Institute. There, they showed us multiple things that astonished me, like neuroplasticity research through giving mice stimuli and seeing their neural reactions, or the first project in the history of the entire world to map an entire complex brain. In projects like these, animals have pioneered the field of neuroscience research, but most of the research is manual labor, data collection, and that takes a lot of time. For example, they told us how to get a better understanding of mammalian brains, they would need to map an entire mouse brain, the next smallest brain that they could do. That process would take 100 years. That is 100 years of neuroscience researchers left in the dark, researching purely off, most of the time, manual labor. There's many problems like these that make neuroscience research a slow, tedious, and laborful process. Hello, my name is Rihanna Patel, and I'm going to tell you how I developed a system that's able to accelerate the next century of neuroscience research by making these problems autonomous. For the purpose of this project, this ball is considered as any animal, mostly transgenic mice, that's being used in neuroscience research. And this robot is used as the hardware that I've programmed my software into to use these 8 infrared sensors as its inputs. Simply, my software can track behavioral, momentum, and stimuli-seeking patterns within the animal as it moves through 4 stimuli zones. It then provides these patterns as an output with over 70 different possibilities, the possibilities only increasing as you get more accurate. Whenever the animal is interacting with the stimuli, my software can autonomously track it, removing the need for hours and hours of manual labor, accelerating neuroscience research. Then the behavioral patterns outputted can be contrasted with other neural data on the subject. This is a flowchart of how my system works. Other than the timestamps, my software works on 3 main functions that produce the 3 outputs that analyze the data. First, the proportion of each stimuli zone visited. It works by finding the vector of the subject through a function I developed, and then comparing that vector to the position of each stimuli. Each IR sensor has its own vector or array with the magnitude and the direction of the subject. I then convert each vector to a coordinate. Averaging all those coordinates, I get a singular vector in the direction of the subject. These vectors are useful for another use of my software, as the hardware orbits around the subject, another test used in neuroscience. To find the behavior pattern of the animal, the software analyzes 3 trials with over 64 different outputs. To examine long-time behavior patterns, I first have to set up an array of every stimulus exhibited and at what timestamp. The behavior pattern either detects constant switching between stimuli in the animal's environment or a repeated behavior around a certain stimulus. These concepts can be connected to various research efforts in neuroscience. Similar to the previous, momentum patterns also call on the last few entries in the array. However, the momentum patterns find the last 2 different entries and get their timestamps to analyze whether the subject is focusing on a stimuli, constantly and rapidly switching around, or avoiding certain stimuli. I know that this project can have far-reaching implementations within automizing neuroscience research and stopping animal cruelty. It's live and let live. The animal can interact with its environment and without interference and still be contributing to cutting-edge neuroscience research. Like any type of engineering, software engineering is a highly iterative process, and I did that greatly, starting from just identifying what stimulus the subject was in to tracking momentum and behavior patterns, and I look forward to keeping on iterating. My next steps would be to create a device that automates the neural research that's taken from the subject, as well as creating an AI-type platform to integrate these 2 softwares. I think as our society, our next steps are to continue the automation of autonomous research, to condense not just the next decade, but the next century of technological innovation for a better humanity for all. Because at the end of the day, innovation is everything. Thank you.